In this video, I'm going to show you the dynamics of muscle contraction under two conditions. One, in laboratory experiments, and two, how it works in our living body. And so I just want to clarify that from the start because a lot of what you talk about with muscle contraction, like a muscle twitch, wave summation, and tetanus, are laboratory conditions. These are conditions that we can see in a laboratory through experimentation that gave us the knowledge to understand how muscle works compared to the way that muscle works in a living person. To start, we're going to take a look at the lab experiments, and I'll show you what a muscle twitch is, and then build on the muscle twitch to show you wave summation, incomplete tetanus, and tetanus. So the lab experiments that I'm referring to here are usually done with a frog. They usually dissect out the sciatic nerve that attaches to the gastroc. So understanding this is an experimental setup, does it make sense that we could use a device to electrically stimulate either the sciatic nerve or we could stimulate the gastrocnemius, the muscle itself? We could also attach the gastrocnemius, cut at the Achilles tendon, to a device that would sense the force of muscle contraction. This is the typical type of setup that's used in the experiments that we're going to talk about. Okay, let's see how this experiment works. In the top graph, what they're doing is they're showing us the amount of voltage that we're delivering to stimulate the muscle. And so in this graph, I can see that what they're doing is they're slowly increasing the amount of voltage that's given to the muscle, and then we're seeing what happens as a result. And so in the picture below, they're just giving us a depiction of what the muscle response is. We'll take a look at that. And then in the third graph, what they're doing is they're showing us the response of the muscle. Remember, we said we would hook this muscle up to a device that would show us how much tension is developed, and that's what they're showing us in this third graph. They're just showing us that when we give a stimulus, what is the strength of the contraction that was generated. Okay, so let's take a look at how this experiment works. In this experiment, what they're doing is they're just varying the amount of voltage. Okay, and so the variable here is they're going to increase the amount of voltage and see what happens. So I can see at time 1, we give a small voltage and it looks like no muscle fibers are contracting. And when I look at the myogram, the amount of tension developed, I see it's a flat line. And so at this low level of voltage, there's no response from the muscle. So let's dial the voltage up. Here, we're increasing the voltage a little bit, and so let's see what happens. According to this picture, it looks the same. Looks like no muscle cells are contracting. And when we look at the myogram, same thing. Zero tension is developed. And so these two stimuli did not evoke a response from the muscle. So let's try it again. Dial the voltage up, and this time what we're seeing is that when we turn the voltage up slightly, that the muscle did respond. So I can see some cells that are drawing more darkly in the muscle that are responding. And when I look at the graph, I can see that some tension was developed. So I want to pause this experiment right here. We'll come back to this, but I want to pay attention to this little contraction that we're seeing on the myogram. This is what's known as a twitch. A muscle twitch is a single contraction in response to a single threshold stimulus. And what we're seeing on the top graph, and they're showing us this with the dotted line, is the stimulus has to be sufficient in order to elicit a response from the muscle. And so any voltage that's lower than what we call threshold will not elicit a response, but as soon as we dialed up the voltage to the threshold level, it did elicit a response. And that single response to the single threshold stimulus is what is known as a muscle twitch. So let's take a closer look at just that. This graph is taking a look at a muscle twitch, a single muscle twitch. And so we can see on the x-axis, time is going by, measured in milliseconds. And so This is a very quick event that we're looking at. And then on the y-axis, we're looking at the amount of tension being developed. So this is a myogram, same thing that we were looking at just a second ago. And so we can see at time 0 that they're giving a single threshold stimulus. And when I look at what's happening, I can see that a few milliseconds are going by where the tension remains 0. This period of time that they're highlighting bright blue on the graph is called the latent period. And the latent period is just this few milliseconds that follow a threshold stimulus where calcium is being released from the SR. And because calcium is being released and binding to troponin, all the molecular events that lead up to contraction, there are a few milliseconds where the muscle doesn't contract. But as soon as cross-bridge formation starts to occur, then we can see that tension starts to increase. And as tension increases, this is what's known as contraction. Okay, so contraction is sliding of the filaments, thin filaments over the thick filaments. Tension is being produced. Once tension starts to decrease, that's known as relaxation. And we can see that it's going to take longer for the muscle to relax than it does to contract, and that's typical. During relaxation, what's happening is the sarcoplasmic reticulum is actively reclaiming all of the calcium, and cross bridges are no longer being formed. It's going back to its resting state. So this is what's known as a twitch. A twitch has 3 phases that you should know. The latent period, when the molecular events of calcium release are happening. The contraction period, when the muscle is generating tension. And then the relaxation phase, when the muscle's tension is decreasing. Here's a graph that compares 3 muscles and the muscle twitch that's seen from a single threshold stimulus. So at time 0, they give the single threshold stimulus, and then color-coded in this picture, we can see that we're looking at a twitch of an extraocular eye muscle, okay, small muscle that moves the eyeball, the gastrocnemius, which is the big calf muscle, and soleus, which is also a big calf muscle. It's right underneath the gastroc. So what should we notice from this graph? I noticed that the latent period is the same regardless of the muscle. So whether we're looking at a small muscle or a big muscle, it's going to take the set amount of time for calcium to be released, and it looks like that's the same from muscle to muscle. But one difference that I'm seeing is that the amount of time that it takes for a muscle to twitch varies depending on the muscle. So I see that the lateral rectus muscle in blue reaches peak contraction and relaxation much quicker than the gastrocnemius, which takes longer to develop peak tension. And then soleus, I see that that muscle takes even longer to develop peak tension. And so depending on the muscle you're looking at, the amount of time it takes for the twitch will vary. A single threshold stimulus will result in a muscle twitch, and muscle twitches of various muscles take different amounts of time. So now that we know what a muscle twitch is, let's go back to this experiment and see what happens. And so in this experiment, what we were looking at is we were looking at just varying the amount of stimulus. And so we're looking at dialing up the voltage and seeing how the muscle responds. And we know that a muscle twitch is just that single contraction due to a single threshold stimulus. So what about this stimulus, the next one that goes above threshold? We can see that the muscle is responding. Actually, more cells are responding. And when we look at the myogram, I can see that more tension is developed. And so let's keep going with this. So we give another stimulus. What would happen if we increase the stimulus? More cells respond, and more tension is developed. This is what's known as recruitment. And so as more electrical activity stimulates that cell, it recruits more motor units and on the job, and the amount of tension goes up. And so this will continue to happen, not forever. So here we can see we increased the voltage. We got an increased response. It looks like all of the cells are contracting, and therefore it would develop a maximal force. And so taking a look at what would happen if we increased the electrical stimulation? Well, when we increase the electrical stimulation, all that can happen is that all of the cells can contract. And when all of the cells can contract, I get that max force generated or max tension generated. And so I see that even if we, turn the voltage all the way up, that all that could happen is that all of the cells can contract, and that's going to generate the maximum amount of tension. And so what I'm noticing in this experiment is that there has to be a stimulus that's threshold in order for the muscle to respond. And once you hit a threshold stimulus, once you increase that stimulus, that the tension developed in the muscle will also increase until we hit a maximum amount of tension. Then, even if the stimulus increases, we can only physically produce so much tension in the muscle. And that amount of tension that's being produced is proportional to the number of cross bridges being formed. And so because there's only a certain physical number of cross bridges there and can be formed, we're going to get a max amount of tension that can be generated. Okay, so far we've looked at lab experiments and we've looked at the muscle twitch. Stay with me, I'm going to pick up the pace and show you about summation and tetanus next. The experiments that we just looked at varied the amount of voltage that was being given. So every time we stimulated a muscle, we turned the voltage up. In the next few experiments we're going to discuss, instead of varying the amount of voltage that's given, we're going to keep the amount of voltage constant, and instead we're going to vary the frequency that we give the stimulus to the muscle and see how that affects contraction. Let's take a look at this myogram. In this experiment, we give a stimulus, we see a twitch in response to that stimulus. I can see that it reaches peak contraction, it's entering relaxation, but notice that before it relaxes completely, we're going to give it another threshold stimulus. The stimulus is the same. That's why they're trying to draw the size of the red arrow the same. We're just giving that stimulus prior to the twitch being finished. So what we see happening is the muscle responds to that second stimulus. And we see another twitch, and the peak tension developed here is even more than the peak tension that was developed with the first. And so let's try this again. Before that twitch comes down to full relaxation, let's hit it with an identical stimulus. Will it respond? Yes, it does respond, and the tension being produced is even greater. This is a phenomenon known as temporal summation. It's also known as wave summation, and it shows us that if the muscle is given another stimulus prior to full relaxation being achieved, it will still respond. Next experiment, let's increase the frequency. And so here we're giving identical stimuli even closer together, and what we see is that the muscle contracts and that it stays contracted. It starts a relaxation phase, but if it gets hit with another stimulus, it responds. And here you can see the plateau at the max tension. This is known as unfused tetanus or incomplete tetanus. If we give stimuli so frequently that the relaxation phase of a twitch is completely eliminated, which is what we see here, what we see is that the muscle will contract. It will stay in a smooth, sustained contraction until that stimulus is removed. This is what's known as tetanus. And complete tetanus or fused tetanus, where we see that constant tension across the top, is a laboratory condition. And now we're on to contraction within a living organism, the way our muscles contract, which are isotonic and isometric contractions. An isotonic contraction is a contraction where the muscle moves the load, like the example that they're showing here. In this example, they're putting a 3-kilogram weight on the muscle. They're stimulating the muscle to contract, and when the muscle contracts, it moves the 3-kilogram weight. Or if we take a look at the graph, what they're showing us is that the tension that's developing within that contracting muscle must equal the resistance in order to move the resistance. And so the amount of tension being generated in that muscle is also 3 kilograms, and that's why it can move the load. This type of isotonic contraction is known as a concentric contraction, and a concentric contraction is when the muscle shortens, compared to another type of isotonic contraction called eccentric. In eccentric contraction, what happens is the muscle lengthens, and it lengthens while it's maintaining tension. So in other words, for this muscle to set down the 3 kilogram weight would be an eccentric contraction. The other type of contraction we see in the body is called isometric. Iso, equal, metric, measurement. That is when the muscle develops tension, but the measurement does not change. So in this example, we can see that they doubled the weight. They're putting a 6 kilogram weight at the bottom of the muscle. Muscle contracts, and muscle is not moving the weight. The weight is too heavy. So why is this happening? As they're showing us in this graph, when the muscle contracts, we can see that the maximum amount of tension that's being developed in the muscle is under 6 kilograms. And so the amount of resistance, the weight that they put on the muscle, is 6 kilograms. Because the muscle is unable to generate that type of tension, it's unable to move the load. That's what's known as an isometric contraction. Keep in mind, the force of contraction, or the amount of tension developed, is directly proportional to the number of cross bridges formed. And so it has to do with the size of the muscle. And so if that muscle can't develop that amount of force, then it can't move the load.